first of all, we said we're gonna start with the action potential traveling along the motor neuron. The motor neuron sends an action potential, same thing as nerve impulse, okay? Action potential. This is that electrical current that travels down the axon, and when it gets to the end of the axon, we said that acetylcholine is released. What is acetylcholine?
Neurotransmitter.
It's a neurotransmitter, right? So here we have the synaptic nod, right, at the end of our axon. And here's our axon, here's our synaptic knob or synaptic bulb, has different names. And here's the little vesicles containing acetylcholine, right? This would be our muscle fiber. Okay, so again, here's the neuromuscular junction. So when the action potential gets to the end of the axon, acetylcholine, stored in the little vesicles here, is released. So it's going to go across this space. What is this space called? Synaptic cleft, exactly. So it goes across the synaptic cleft and it binds to receptors here on the motor end plate. Okay, so remember this kind of folded up part of the sarcolemma is just a special receptor site for acetylcholine called the motor end plate. Okay, so it binds to receptors on the motor end plate, okay? And that is what will then trigger an action potential in the muscle fiber. So when acetylcholine binds here, it will kind of cause this this different event. So the reason why I say that is because I don't want you to think acetylcholine is traveling along the membrane. It's not acetylcholine. Acetylcholine just binds to its receptors. But that binding of acetylcholine causes little channels to open in the sarcolemma. And remember we said that the action potential is actually ions, atoms that have an electrical charge crossing over the cell membrane. And that's exactly what happens. So when acetylcholine binds, the little ion channels open, allowing it's actually sodium and potassium, and you'll talk about that more when you take physiology, to cross over the cell membrane. And that is what the action potential is. So the action potential will spread in both directions along the sarcolemma, and it will go down the T-tubules. Remember, they carry the action potential down inside the cell. Okay. Remember that on either side of the T-tubules, we have the little sacs called terminal cisterns. Terminal cisterns store calcium. So when the action potential goes down the T-tubules, the terminal cisterns here are going to release calcium. Right, so we have terminal cisterns. My terminal cisterns are very weird shapes. Okay, so they go, that will allow the calcium to go out into the sarcoplasm. Okay, when calcium is out here in the sarcoplasm of the muscle cell, what does it bind to? You guys know this. It binds to a molecule on the thin filament, right? Troponin. Okay, so that is the next step. And by the way, one more thing here. I just want to clear up in case this is confusing. A muscle impulse, again, same thing as saying action potential. It's just another name for it. Okay? So you can think of all of this as one thing triggering the next, right? The electrical current or action potential traveling along the axon triggers the release of acetylcholine. The binding of acetylcholine to the motor end plate triggers that electrical current in the muscle fiber. As the electrical current or action potential travels along the sarcolemma, that triggers the release of calcium, right? Okie dokie. So calcium will bind to troponin on the thin filament. Remember when calcium binds to troponin, because troponin is right next to the tropomyosin molecule, that will kind of shove the tropomyosin over, right? Kind of shifts the tropomyosin, and these binding sites on the actin molecules will be exposed. When these binding sites are exposed and they're just kind of hanging out there, what is going to bind to them? What binds to actin? Myosin, right? Myosin on the thick filament. The myosin heads are just kind of waiting for those binding sites to be exposed. So the myosin heads bind to actin, and they have that swiveling motion. So that's what, as the myosin and actin bind, it pulls the thin filaments toward the center of the sarcomere, so the sarcomere shortens. Okay, so that sliding of the filaments is what contraction actually is. That's what causes contraction. So this really is not that many steps. Okay, and so even now, if you feel like, as we're looking at this, if you're feeling like, oh my gosh, I'm totally lost, you can get this by Wednesday. You guys have enough time to get this down, okay? So remember, as you're going through the structure of the muscle cell and you look at the diagram showing the parts of the filaments like the troponin, the tropomyosin, just go through their basic functions, and that will kind of walk you through these steps. Okay? Or as you go through these steps, it kind of makes you kind of review the parts of the muscle cell. Any questions on any of that? Okay, so this is what happens to cause contraction. Now, everything kind of just reverses when we want to relax. The motor neuron stops sending action potentials, so that will stop the release of acetylcholine. That stops the action potential in the muscle fiber. Okay, so everything just kind of stops. But what happens when we have this chemical that's released here, right? Acetylcholine is a chemical. So when it's released, some of it will bind to its receptors, but we still are going to have some acetylcholine kind of hanging out in the synaptic cleft. It will continue to bind to the receptors here, and muscle contraction would continue, kind of out of our control. And we, of course, don't want that to happen. We want to have precise control over our skeletal muscle. So what happens is that although some acetylcholine will bind to its receptors, and that will cause an action potential here, the rest of it is immediately broken down by an enzyme that is in the synaptic cleft. So this is what allows us to have really precise control. If we want to continue that contraction of the muscle or have another contraction of the muscle, then we have to have another action potential. Okay, and so everything has to happen again. So this is one thing that has to happen to allow the cell to relax. Acetylcholine is broken down here by an enzyme. The other thing is, same thing with calcium. Calcium is a chemical that's released out into the sarcoplasm of the muscle cell. So whenever it's released, some of it binds to troponin, but we don't want it to continue binding, because our muscles would be contracted, we wouldn't be able to relax. So to prevent that, any excess calcium is pushed right back in to the sarcoplasmic reticulum. And so we have what are called calcium pumps that are actually in the membrane of the sarcoplasmic reticulum, and they keep moving the calcium back in. They're very efficient. They actually can store 2,000 times more calcium inside these tiny little sacs than we have out in the sarcoplasm here. So they push the calcium back in very quickly. So those two things are constantly happening. Whenever we have the release of acetylcholine, it's broken down right away. Calcium is pumped back into the terminal cisterns of the sarcoplasmic reticulum, like as soon as it's released. So that will allow the cell to relax, okay? And I think that those things are mentioned here, I believe, somewhere. If you go through this, I think it will talk about that. I know your book talks about these two things, but I think it's actually in this diagram. So this is what we just talked about. So acetylcholine is broken down in the synaptic cleft by this enzyme called acetylcholine esterase. Long word. This is how it's abbreviated. So I think of this as like erasing acetylcholine esterase, and I would like you to know the name of that enzyme. And then calcium is also, as soon as it's released, the excess is pumped back into the terminal cisterns by what are called calcium pumps. A pump means that it requires ATP from the cell. It requires energy. And this is actually how muscle cells use a lot of their ATP. They actually use a lot of it just to move the calcium back into the SR so that the cell can relax. So actually relaxation requires quite a bit of energy. You wouldn't really think that, but it does. Okay, do you guys have any questions on that? That actually is just about it as far as muscle tissue. Okay, and that's everything. So we are pretty much done with the muscle tissue. This is just something I wanted to talk to you guys a little bit about. And I think I may have mentioned this. Did we talk at all about muscle names? Okay, so we'll come back to that in just a second. You guys have any questions, or anything you can think of as far as those steps leading to muscle contraction, neuromuscular junction?